So today we're talking about gene regulation. Last time we talked all about how you're going to create a polypeptide, how you create a protein. You take this gene encoding region of DNA, you transcribe a piece of RNA, that RNA is going to be translated to a ribosome. Based on what I told you in the last lesson, this should just keep going and going and going forever and ever and ever all the time. But the thing is, we know that doesn't happen. We control, or our body controls, what proteins are being made at any given point. And usually the way they control it is through gene regulation. Now, I don't know if you guys remember, but a long time ago we talked about biochemical pathways. Do you remember that? We talked about steps in the lactose, like how lactose is broken down. I said I'd step out and then come back, yeah. We talked about rate-limiting steps, things like that. We talked about cellular regulation, genetic regulation, and biochemical regulation. Does anybody remember what I'm talking about? Vaguely remember it because it was on the last exam, or 2 exams ago. It was a long time ago. So we're revisiting that. So if you remember it, great, you'll have an edge. If you don't remember, don't worry, I have a whole lecture about it. I know. So we're really just focusing on genetic regulation, controlling how that gene is expressed. This lets different levels of genes get expressed under different circumstances. The Arctic fox. Check this out. In the summer, it's brown. In the winter, it's white. Yeah, isn't that cool? It knows it's able to transition, not like consciously, but due to environmental cues, whether it's the length of the day or the temperature, you're going to go from producing pigment to not producing pigment. It's pretty easy. You just turn off the genes. Turn off the genes making that reddish color, and suddenly all of the fur goes white. Suddenly, it's like poof. Where did it go? No, but it takes some time. But the genes turn off, and the color fades gradually, and then right. Yes. It's totally a defense. It's all about camouflage. Right. So by controlling that pigmentation gene, this organism is able to give itself a reproductive advantage. A guy named Jacob and Monod, 2 guys, sorry, they noted that cells don't produce enzymes that break down products until they're exposed to that product. That is, if you guys eat a lot of sugar, you're going to produce the enzymes you need to break down that sugar. But if your body's not exposed to sugar, you're not going to produce those enzymes. You only produce the stuff that you need to break down whatever you're eating at that point. Does that make sense? No, this is called bioregulation. Otherwise, you're going to constantly be producing molecules that you don't need. I have no idea how the body works. I have no idea. It's anatomy. I don't do anatomy. I do genetics. I took anatomy, yes. It was terrible. And I said, I will never do this again. And I didn't. This is called enzyme adaptation. When suddenly, once you're exposed to those that product, you can start to break things down. They figured this out by looking at bacteria, because bacteria are really easy. They grew this bacteria on a sugarless plate. Sugarless. There was no sugar on the plate whatsoever. And they checked what enzymes were being produced. Not many sugar breaking down enzymes. And then, did they still find some? They found the enzymes that were active were the enzymes that were needed to break down what was in the media. What they were feeding it. Right. And then they switched food. And right. It didn't die. It started producing the enzymes it needed to break down that product.
Actually breaks down its own enzymes, recycles them,
and then swings it to a new product. Life is awesome. Yeah. That's called enzyme adaptation. For the longest time, they won the Nobel Prize for this. This was big news. This started making us think about how life actually works, how we actually function, what's going on deep down in our cells. No, well, we'd have to have the genes, in order to break down plastic, you'd have to have the genes necessary in your body already to do that. Now, back to CRISPR-Cas, then yeah. What about dogs?
Yeah, like they eat all types of crazy.
They do, but it doesn't mean they can break it down.
It's like how people are lactose intolerant.
Right. Some people are lactose intolerant because their lactose, beta-galactosidase doesn't work. So if we can control the genes, we can allow for different levels of expression at different times under different circumstances. And there's a lot of ways you can control a gene. You can control it through transcriptional control. With transcriptional control, you're saying you're effectively allowing it to build RNA. You can control it through RNA processing. You make that poly A tail really long, or you give it a different cap. You change the expression. You can control it through translation. You speed up or slow down the ribosome. You speed up or slow down production of the polypeptide. So you can control what enzymes are present by controlling the RNA or the DNA. That's genetic regulation. This has enormous benefits. Like I said, you're not wasting energy. You don't want to waste energy. You know, building these things takes a ton of energy. You're building, whether you're building RNA or a polypeptide, you're building single and double bonds. You're investing a lot of energy in those bonds. You don't want to waste your time or waste your energy. So by regulating gene expression, you only make the proteins when you need them, not just willy-nilly all the time. Now, there are some genes you might make all the time. Maintenance genes. It's called constitutively active gene products. But a lot of times, those that are on all the time are there in order to stop other genes that are more energetic from being made. It's sort of like, I don't know, if you leave a night light on, it means you don't have to have the overhead lights on. So you're going to have a lot less energy being consumed. There you go. Yeah, if you have a security guard who can call it in when they're needed, you don't need to have 20 police officers around. So you can control a lot of things genetically, and we have talked about all of these. Metabolism, cell division, responding to the environment. Each of these processes is going to be controlled ultimately at the genetic level. With metabolisms, you have some proteins that break down other proteins. So if you control enzyme production, you can control reaction rates. That's one way you're going to control how much of a particular substance is in your body at any given point. You don't want to have so many enzymes that you wipe out your body of, I don't know, sugar. You want to have some hanging out in there. You don't want to have your body, absolutely none. So you want to have a median level of enzymes. You want to have enough that you're still conducting your cell's metabolism, you're still keeping yourself going, but not too many that you're entirely wipe out or deplete your system. You have things that will help you react to environmental stresses. You have these things called heat shock proteins. So you guys know, when you get a fever, it's bad, right? Okay, your body temperature goes up. What happens to enzymes at a high temperature? They denature. And when they denature, they stop functioning, and that's permanently stop functioning. So why is it that when you have a fever, you don't die?
Because other things kick in.
Other things kick in. You have these things called heat shock proteins. You have proteins in your body that will sort of almost act like a fireman's jacket. They wrap around the protein in order to stop it from getting too hot and maintain its shape.
So it protects it when you have a fever.
Yeah, it protects you when you have...
And so it keeps working once your fever goes down.
Exactly.
Is that why you break into a sweat?
You break in a sweat, actually, in order to drop your body temperature.
I thought you were mostly sweating to remove the toxins from your blood.
No. Sorry. I couldn't tell if you were being serious or not. No, you don't sweat to remove the toxins from your blood. You sweat because it cools your body. You remove salt from your body. You sometimes. If you got a urea buildup that big that's in your sweat, you got issues. Well, you know what? Not good at anatomy, so I'm pretty sure that your kidneys are what remove urea. That's the big thing. That's why you have urine coming out. It's not just bacteria, but that's because, okay, now we're talking about your microbiome. That's Bio 102. You sweat out, you do sweat stuff. But for the most part, it's salt and water, yeah. Salt. Let's talk about salt. There we go. Salt's on the screen. Salty environments. Why would it be bad for you to be in a very, very salty environment? That's right. Why does it dry you out? What process is happening there? Hypertonic, hypotonic, isotonic. Okay, what's the process, though? What do you mean you have no idea? It doesn't matter. It's the same process occurring. Broad process name. No. No. No.
Combustion.
No. What does it start with? Oh,
You don't even know.
no, it starts with an O. Osmosis. It's like, oh, you're like, oh, I was just working on that exam thing. It's one of those words, major important thing for biology. Osmosis. So, if you're placed into a hypertonic solution, your water is going to leave. The water in your cell is hypotonic, and water always moves from hypo to hyper. So your cell is losing water like crazy. When your cells get to a certain point and they lack water, they crenate. They die off. Deal is there are going to be some proteins in there, some proteins that you need that will protect the vital parts of your cell from dying entirely, that will protect your nucleus. And you only need those when you're exposed to a lot of salt or extreme dehydration. Oh, okay. Cell division. You absolutely have to control cell division. What are we constantly doing during cell division? What happens 3 times in cell division? Yeah, okay. Okay, I'll give you a hint. It happens during G1, it happens during G2, and it happens during metaphase. Checkpoints. You're constantly checking yourself out. If the cell is able to reproduce, is viable, if it can survive, it's going to reproduce, but it's only through those checkpoints. What those checkpoint genes do is they hit the DNA and they will turn on a specific protein. They're going to turn on the production of a protein, and that protein, that cyclin, is then going to control a lot of other things by turning on or turning off the DNA. It's all about genetic regulation. You can turn things on and turn things off, and then they will build up to high volumes or drop very quickly. So like I said earlier, we have 4 levels of gene regulation. Transcriptional control, RNA processing, which is where you're going to modify the RNA. Translational control, we're going to interact with the ribosome. And then post-translational modification, things you're going to change to that protein after it's been produced. Yes?
So, we control which genes.
Mm-hmm.
Do we do it knowingly, or does your body?
Your body just does it. You have no idea how this works. Okay. Well, we know how it works, but your body doesn't control it. It's a reaction to the environment. It could be a reaction to you eating something. It could be a reaction to you breathing something, to you being in a certain temperature. Transcriptional control occurs if a molecule directly interacts with the DNA, turning off the transcription of the RNA. This regulates the final gene product. So you're coming into the cell, or you're having an effect on the cell that directly connects with the DNA, not some later step. RNA processing control occurs when an RNA molecule gets processed after being transcribed. So you'll get a polyadenal tail, and if you remember, a poly-A tail means that the RNA transcript can last longer in the cell. Well, if you have a shorter poly-A tail, that will control how much gene product is being produced by that RNA, because it only exists for so long. Or you might splice the exons and splice the exons together in a different order, leading to a different protein or a different function of that protein. Translational control is connected to the efficiency of RNA translation to a protein. You could have small molecules that could either speed up or slow down transcription by helping the ribosome bind or by preventing the ribosome from binding to the RNA. And finally, you've got post-translational modification. After the polypeptide is created, it can be modified. Proteolysis and glycosylation are going to help form a mature protein. Now, proteolysis, as you remember, is where you're going to cut the polypeptide into pieces. Obviously, the smaller piece has a different function than the larger piece. By adding carbohydrates with glycosylation, you will slightly change the function. All of these are ways of regulating the function of a protein.